Hi everyone, Neetu here. I hope all of you are staying safe at your home. We'll start with our biochemistry classes. Today we'll discuss about chemistry of carbohydrates. So how will you define a carbohydrate? They are polyhydroxy, aldehyde or ketone or compounds which yield these derivatives on hydrolysis. They are composed of elements carbon, hydrogen and oxygen. So saccharide, it is a Greek word which means sugar. And what are the functions of carbohydrate? It is the most abundant molecule and it is the main source of energy in the body. Brain cells, RBC, they are almost fully depend on carbohydrate as their energy source. Then it is the storage form of energy. That is energy, it is stored as starch and glycogen. This excess carbohydrate that can be converted into fat. Then we have glycoproteins and glycolipids. They are also components of cell membrane and receptors. Then the structural basis of many organisms like cellulose of plants, exoskeleton of insect, cell wall of microorganism, mucopolysaccharide as the ground substance in higher organisms. Depending on the number of monomer units present in carbohydrate, they are classified into monosaccharide, disaccharide and polysaccharide. Monosaccharides, they are the simple sugars which cannot be further hydrolyzed into simple form. Examples are glucose, fructose, galactose, etc. And when two monosaccharide units are combined together, it is known as disaccharide. Examples are maltose, sucrose, lactose. Oligosaccharide means they contain 3 to 10 monosaccharide units which is joined by glycosidic bond. Then polysaccharide, they contain more than 10 sugar molecules. Example, starch, glycogen and cellulose. As we said, monosaccharide, it is the simplest sugars which cannot be hydrolyzed further into simple compounds. What are the examples? Examples are ribose, glucose, fructose, galactose, etc. Glucose and fructose, they are the most abundant monosaccharide in nature. This monosaccharide, they cannot be hydrolyzed into simple compounds again, but depending on the number of carbon atoms present, they can again be classified into triose, tetrose, pentose, hexose, etc. If 3 carbon units are present, it is known as triose. Example for triose is glyceraldehyde. Then 4 carbon atom, it is called as tetrose. 5 carbon atoms, it is called as pentose. Example for a pentose sugar is ribose. Then if 6 carbon atoms are present, it is known as a hexose sugar. What are the examples for hexose sugars? Glucose and fructose. Monosaccharide, they can also be classified on the basis of the functional group present. Aldoses are monosaccharide which have an aldehyde group and many hydroxyl group. Such sugars are known as aldoses. And ketoses, they have a ketone group as the functional group. So monosaccharide, they are the simplest form which cannot be further hydrolyzed. They can be classified on the basis of the functional group present. That is, if an aldehyde group is present, that is known as aldose sugar. And if a ketone group is present, that is a ketose sugar. Example for aldose sugar is glucose and ketose sugar fructose. And monosaccharide, depending on the number of carbon atoms present, they can be classified into triose, tetrose, pentose, hexose, etc. Now we will see the general properties of monosaccharides. So first we will discuss about isomerism. So what are isomers? They are compounds with similar structural formula but they differ in their spatial arrangement. And this isomerism, it is due to the chiral carbon. They differ in their configuration, that is in one of the carbon group or more than one carbon atom or with different conformation or with different physical properties. Asymmetric carbon atom or chiral carbon atom. That is, a carbon atom is said to be chiral or asymmetric when 4 different atoms or chemical groups are attached to it. So the simplest monosaccharide with an asymmetric carbon atom is glyceraldehyde, and this is taken as the reference for naming higher carbohydrate. So if a compound is having one asymmetric carbon, it may exist in two forms, which are the mirror images of each other. And the number of possible stereoisomers, it depends on the number of asymmetric carbon atoms. So glucose has 4 asymmetric carbon atoms, that is the second, third, fourth and fifth carbon atom. They are the asymmetric carbon atom. So the number of possible stereoisomers for glucose is 16 isomers. So as we said, stereoisomers, they can occur either as D or L form. How do we call it as D or L form? This is based on the position of hydroxyl group on the reference carbon atom. So in glyceraldehyde, which is the reference carbon atom, carbon 2 is the reference carbon atom. So if the hydroxyl group is on the right hand side of the asymmetric carbon atom, then it is known as D-glyceraldehyde. And if the hydroxyl group is on the left hand side of the carbon atom, then it is called as L-glyceraldehyde. And in glucose, which is the reference carbon atom or penultimate carbon atom, that is the C5. The reference carbon atom in glucose is carbon number 5. So this is the reference carbon atom. Carbon number 5, 1, 2, 3, 4 and 5. So carbon number 5 is the reference carbon atom in glucose. So if the hydroxyl group is on the right hand side, then it is known as D-glucose. So here you can see hydroxyl group is on the right hand side. So it is known as the D-glucose. Here this is the reference carbon atom and the hydroxyl group is on the left hand side. So that is known as L-glucose. So what is the significance of D and L forms? Remember, it is capital D and capital L. So majority of the monosaccharide which is present in mammals, it is in the D configuration. That is, the hydroxyl group is present on the right hand side of the reference carbon atom. Except L-cellulose, which is a metabolite of uronic acid pathway, then L-fucose, L-deoxygalactose, they are constituents of glycoproteins. So in mammals, the enzymes of Next is optical activity. So all the compounds having an asymmetric carbon atom will exhibit optical activity. That is, when a beam of plain polarized light is passed through sugar solution, then the light will be rotated either to right or to the left. If the light is rotated to the right, that is in the clockwise direction, then the molecule is said to be dextrorotatory. It is represented as small letter d or with a plus sign. And if the light it is rotated to the left side, that is anti-clockwise direction, then it is said to be levorotatory. It is represented as small letter L or with a minus sign. And you should not be confused with the previous thing, that is D and L forms of stereoisomers. D and L forms, it is represented by capital D and capital L. And this optical activity, that is dextrorotatory or levorotatory, it is represented by small letter D or small letter L. So glucose, it is dextrorotatory in nature. That means it can rotate the beam of plain polarized light to the right side. And fructose, it is levorotatory. And a mixture containing equal quantities of dextrorotatory and levorotatory, then it is found to be optically inactive, and this is called as racemic mixture, and it is represented by DL mixture. Next is epimers. So isomers that differ from one another in their configuration around one specific carbon atom only, then it is known as epimers. So when two monosaccharides, they differ from each other in their configuration with respect to a single carbon atom other than the reference carbon atom, then they are called as epimers. That is D-glucose and D-mannose, they are epimers, and that are formed due to interchange of orientation of OH and H around carbon atom 2. D-glucose and D-galactose, they are epimers that are formed due to the interchange of orientation of OH and H around carbon atom 4. Glucose and mannose, they are epimers, and they differ from each other in configuration with respect to a single carbon atom other than the reference carbon atom. So the difference between glucose and mannose is here. So you can see the orientation of H and OH group in mannose and H and OH group in glucose. So, D-glucose and D-mannose, the difference is only at the second carbon atom. Then D-glucose and D-galactose, they are also epimers, the difference is at the fourth carbon atom. So, you can see the orientation of H and OH group in D-glucose and H and OH group in D-galactose. So, which are the epimers of glucose? The epimers of glucose are D-mannose, the difference is only at carbon 2, and D-galactose. In D-galactose, the difference is at carbon atom 4. So, the epimers of glucose are D-mannose and D-galactose. Next is anomerism. Sugars in solution, they exist in the form of ring. The ring is formed because of the high reactivity of the carbonyl group of both aldehyde and ketone. So, in aldohexosis, a hemiacetal ring, it is formed between the first carbon and fifth carbon. So, here the hemiacetal ring is formed between the first carbon and the fifth carbon. So, this results in a ring form containing 5 carbon atoms and 1 oxygen. And this structure, it is similar to a compound known as pyrene. So, this is known as the pyranose structure. The formation of ring structure makes carbon 1 also asymmetric. So, carbon atom 1 shows alpha or beta form depending on the position of hydroxyl group at carbon 1. So, if the hydroxyl group on the anomeric carbon atom extends to the right, then it is known as alpha anomer. So, here the hydroxyl group on the anomeric carbon, it extends to the right. So, it is known as alpha anomer. And in beta anomer, the hydroxyl group on the anomeric carbon atom, it extends to the left. So, here you can see the hydroxyl group on the anomeric carbon atom extends to the left. So, that is known as beta D glucose. So, we have discussed about the chemistry of carbohydrate, that is the classification. Carbohydrate, they are classified into monosaccharide, disaccharide and polysaccharide. So, monosaccharide, they are the simplest forms and they can again be classified into, depending on the functional group present, they can be aldoses or ketoses, and based on the number of carbon atoms present, they are classified into triose, tetrose, pentose, hexose, etc. Then we have discussed about the properties of carbohydrate, where we have discussed about the stereoisomers, the D and L forms, then optical activity, dextrorotatory and levorotatory. Then we have also discussed about the epimers and anomerism. The remaining things we will be discussing in the next class. Thank you.